Yes, so I am extremely privileged actually to be allowed to work on the finger data set, the finger study. So the finger metabolomic study comprises 1,100 people around that. Half of them go through the finger intervention, which is a multimodal intervention including diet, physical exercise, social contact, and cardiovascular control. So these people are then compared, of course, with the ones that don't go through that process. And then we have they had cognition measured, and we produce a cognitive test that we compare with the metabolites. And these metabolites are obtained using the main platforms in metabolomics, meaning NMR for smaller molecules, and also for a number of lipoprotein measures. That's like around 112 lipoprotein measures. And then we also have LCMS data on the same people, and from there we obtain both semi-targeted data, meaning that we know some metabolites that are there and we measure them specifically, and we also do kind of an untargeted approach in which we measure anything we can, and that means like 11,000 metabolomic features. Now that's just the data. Then when we pose 3 questions, first one being how does the intervention changes the metabolites versus the control, the non-intervention. Okay? And so in that case we found that there was, on the people that went through the intervention, there is an increase of acylcarnitines, which are molecules related with energy production and lipid beta oxidation in the mitochondria. And so these acylcarnitines are shuttling fatty acids into the inside of the mitochondria. And the ones that we found are specifically acylcarnitines with double bonds that come from fatty acids that are unsaturated, not saturated fatty acids. And so the other thing that we found is that there's a reduction in sphingomyelins of numbers of carbon like 15, 17, 19, and in general what's more normal is like the even numbers like 20, 18, 16. And so that should be pointing us to something, to some kind of pathway, because it only affects those type of sphingomyelins. Sphingomyelins are these lipids that make part of cell membranes, etc. So then that was the first question. Then the second question was if the variation in metabolites is associated with the variation in cognition on these people, regardless of the finger or non-finger. Okay. So when we did that, we don't see a massive effect. There is some effects, like on the level of lipoproteins, at the level of small classes of molecules, but it did not become our main analysis because in terms of power, because you need to calculate the delta, you lose half of the samples, kind of. So you lose power for the statistics. The third question was the one that became most relevant, which was if we measure your metabolite levels now, how does those metabolite levels impact your cognition after 2 years? Okay, so having some metabolites at a certain level, is it good or bad for you in 2 years? Okay, and so when you do that model, we got a massive pattern of anti-inflammatory metabolites. Okay, that includes mostly omega-3 fatty acids in all types of, not all types of classes, but in major classes of lipids, say in triglycerides, in cholesterol esters, in phosphatidylcholine, phosphatidylanolamines, etc. And so that pattern is a massive pattern. There's a lot of molecules that are appearing to be elevated, and then people have better cognition. Okay. On the other hand, if you have acylcarnitines saturated ones, so different from the ones from the first question, if your acylcarnitines are increased, you have less cognition. Okay, and so those are the main findings of the study. Is this very strong anti-inflammatory pattern that is good for you, and the pro-inflammatory that is bad for you? Well, we think again that, you know, if you think about this, about which molecules were affected, then you go directly to those patterns. Okay, so we have, for example, energy. Actually, there was also a molecule related to the pathways related to energy and kind of degradation of glucose. And so it also suggests there's kind of a reduction of energy pathways. And the other thing that we also notice, there's also another group of molecules that are methylated molecules, and those molecules are nucleotides types. And so it looks like there is a pattern of methylation also on some molecules. That would mean a reduction of gene expression. Okay. Otherwise, the major pattern that we have there is an inflammatory pattern, and so I don't know exactly which pathway because we didn't yet find the way that we would understand exactly which pathway is relevant for this pro or anti-inflammatory pattern. But the other thing we're also doing is trying to understand which part of the intervention is hitting each metabolite. Is it the diet that is hitting that? So rather than not that we would not like to understand the pathways, but for us it's very important to understand what is each part of the intervention, because it's a multi-domain intervention, is hitting certain metabolites and increasing them or decreasing them. Okay. So again, and to conclude, you have inflammation pathways that we don't know specifically which one, but definitely inflammatory pathways, and you have maybe an increase in methylation and decrease of gene expression, and you have this kind of impaired energy production, because we see those acylcarnitines that are reduced, or when they reduce, you have better cognition.